Welcome to our lesson where I'm going to show you how to prepare an Elodea and a cheek cell slide to view on your microscope in our lab. The first thing you need to do to make sure you're ready is make sure you're wearing your safety goggles so that you protect your eyes, because there are some chemicals that if you get in your eyes will really hurt. The first slide we're going to prepare is our Elodea slide. This is the Elodea plant here. You find it in fish tanks. Some of you might even have it at home. Here is our slide. This is where we're going to put the leaf on. The first thing you're going to do is take your pipette and squeeze it, push it in like that, and put it in to your beaker with the water in it. Make sure you can see it. So here's my beaker with the water. I put it in at the bottom and suck some up into my pipette. Now I'm just going to put one drop of water onto our slide. The rest can be emptied back into our beaker, and I'll put our beaker aside. Now I have my slide with a drop of water on the top. The next thing I'm going to do is take a leaf off the Elodea plant. Just one leaf, you don't need many. Take one leaf off gently, and then you're going to place it on top of the drop of water on your slide. Okay, now we have you see that? We have our leaf on top of our water. The next thing you're going to do is take your cover slip. The cover slips are the little plastic things that look like this. You're going to take your rectangular cover slip and you're going to lay it over your specimen on your slide. Now you don't want to do that by dropping it straight down because you might get a lot of air bubbles. What you're going to do instead is put it on an angle like this, but a 45-degree angle, and let it go down over your slide gently to try and avoid having air bubbles. Now your slide should look like this, where you've got your slide, water, your Elodea plant, and then your cover slide on top. The cover slide is to protect our microscope. Now we're ready to mount it on our microscope. Our microscope, so the first thing you're going to do is make sure it's turned on. You make sure it's turned on by, the switch is along the side here. It's this round one, and you're going to turn it up. Make sure you turn it to level 3 so that it's all the way on. The next thing you're going to do is over here, there's a clip. You're going to pull that clip back, and it will allow us to put our slide on our stage. Then you're going to center it so that your Elodea leaf is in the middle underneath where the light comes through, and close down your clip there. The next thing, these microscopes are a little bit different. So to move where your slide is in the middle, you're going to turn this knob to move it left and right, and the knob at the back here to move it forward and back. You shouldn't need to do it because hopefully it should be near the center, but in case you need to move it. Next, you're going to look through the eyepiece, and hopefully you can see what your Elodea leaf looks like through the microscope. It should look pretty neat. Our microscopes have a coarse adjustment knob right here. If it's not in focus, you're going to turn it a little bit at a time until it's focused. Make sure everyone in your team has a chance to look. Then you're going to turn it to medium power, which is the yellow one on our microscopes, and look through the eyepiece again and look at your cells for the second time. Then turn it on to high power. If you need to adjust the coarse adjustment knob slightly, do so very carefully, and then you'll be able to look at our Elodea leaf through the microscope. On our lab, now you're going to draw a sketch of it on your lab sheet. Now we're finished with our Elodea leaf. You are going to first turn your nose piece of your microscope so that it's on low power, or this objective is facing down, the red one, or 4 times. You are then going to remove the slide carefully by pulling this clip back and removing the slide from the stage. Now we're done with the Elodea leaf. The next one we're going to do is our cheek cell. This is a really exciting one. For the cheek cell, the first thing you're going to do is take a new slide. This is my new one, it's clean. And you are going to take a new pipette, your clean pipette, it looks like this, and you're going to take your pipette and put it into our methane blue and suck up a little bit. You don't need very much of this because it's really strong. You are going to put a little drop onto your slide and empty the rest back in here. Be very careful with this because it can stain your clothes and your hands and the desks. So you need to be very careful with all of it. The next thing you're going to do is take your toothpick, your clean toothpick, and you're going to rub it along the inside of your cheek. You're going to rub it up and down 12 times. So you put it inside your cheek. And really rub it around in there 12 times or until you have a lot of it. Then you take your samples of your cells and you're going to roll them around on top of your slide. Roll them around in there so you get lots on there. Then you're going to make sure that you throw this in the garbage because it's bad for people's health if you leave it around. The next thing you're going to do is take your cover slip and again at a 45-degree angle, you're going to drop it down on top of your sample. Look at the top, trying to make sure there's no air bubbles in there. And now we're going to mount it into our microscope. Make sure before you put in your microscope that your microscope is on low power, and ours is. So now I'm going to slide our slide into our microscope. It's really important that now you check and see if you can see any cells. You should be able to see a few floating around in there. You should see lots of different specks across your screen, through the microscope. Then you can turn it on to, rotate your nose piece, turn it on to medium power, look through, make sure it's focused. And then after that, you can turn it on to high power. And again, make sure it's focused through the microscope. That's what we're going to do in our lab. When you're finished, make sure you turn it back to the low objective, take our slide off, and please make sure you clean all the slides at the sinks on the side before you leave. That's all for now. I'm looking forward to our lab together.